All right, this is a video on some kidney physiology. And so this diagram right here is representing what's called the nephron, or the functional unit of the kidney. And nephrons are composed of a lot of different anatomical parts, but generally they're seen as having a cortical region, or a region that's kind of exposed to the outer layer of the kidney, and a medullary region, which is within the kidney, within the middle of the kidney. And so these nephrons, there's a lot of nephrons per kidney, upwards of millions, and each nephron will have an arteriole that supplies it, and this arteriole is derived from, or it's a bifurcation from, the renal artery. So the renal artery will give rise to a bunch of arterioles that go ahead and form these afferent arterioles, and these afferent arterioles are carrying blood towards this structure right here called the glomerulus, which is a capillary bed, which allows for the diffusion of molecules from this capillary bed into this structure right here. And what this is called is these molecules are moving via diffusion, but fluid is also moving through a hydrostatic pressure gradient because the pressure right here is pretty high. So I've got high pressure within the glomerulus. It's pushing fluid from this space within the capillaries, so within the lumen of the capillaries, into this region right here. And this is all created by an epithelium, but it's a tubular structure that has a direct output to the external environment. So once I cross this epithelium right here, both of these epitheliums, so I've crossed one epithelium here and another epithelia here, so two epithelium, now I have entered a space that's within the body, but considered outside of an epithelium. So once a molecule is within this space, within the tubular space of our nephron, it has direct access to the external environment without ever having to cross an epithelium. So I've got this molecule, it's present here. It'll just cruise throughout this tubular structure called our nephron all the way out until eventually it reaches the bladder and is excreted via urination. And so there's a pressure gradient that's moving molecules out into this space. But this region right here is what's called the proximal convoluted tubule. And the logic is the kidney filters a bunch of stuff out. So it's filtering everything small out, including water. So water is being pushed out, and then small molecules are also being filtered out, including glucose, including amino acids, including lactate, including ions like sodium or potassium or calcium or magnesium or chloride. And the idea is that I'm filtering all this stuff out, and I have selective channels present in this region that can reabsorb the stuff that I want, and everything else that I don't selectively reabsorb will continue out through this region and eventually be excreted from this nephron and eventually excreted from the body. So it's the idea that I'm filtering stuff out and I'm selectively reabsorbing stuff. If I don't selectively reabsorb the thing that's being moved through these little tubules, it will exit the body. And so present on the membrane of these epithelial cells that are located on the proximal convoluted tubule in this region are a bunch of channels that move glucose, amino acids, lactate, ions, everything that's small and biologically important from the space within the lumen, so this space, into the interstitial space. So I'm moving it from a space that's inside the body but outside of an epithelium into interstitial space, so it could potentially be reabsorbed into the peritubular capillaries, or these capillaries that are kind of surrounding this whole nephron loop. And so I'm reabsorbing a bunch of stuff, but let's say I have a molecule that's moving down and it wasn't reabsorbed. It'll continue moving out unless it's reabsorbed elsewhere in the kidney. And so this region right here, so I've moved past this proximal convoluted tubule where there's a lot of reabsorption occurring, moving down into this descending limb of what's called the loop of Henle. So this is the loop of Henle, and it contains both a descending limb, which is this side, and an ascending limb, which is this side. And this is considered the descending limb because molecules will be moving down. It's the movement of molecules, movement of water. And then this is the ascending limb because molecules will be moving up. And so in this descending limb, this region is relatively impermeable to ions. So the epithelia that make up this border, like imagine there's like a bunch of cells right here, epithelial cells that make up this border, are impermeable to ions. So they don't really allow sodium or calcium or potassium across the membrane, but they're pretty permeable to water. And they have these structures present on the membrane of these cells called aquaporins. And specifically this kind is aquaporin 1, which allows water to move from the space within the tubule out into interstitial space. And so these are located on both sides of this descending limb. Imagine these are all epithelial cells. that make up this descending limb. These all have aquaporin 1 proteins present in the membrane. And I believe they're on both sides. I don't know if it's specifically aquaporin 1, but there's definitely aquaporins on both the apical side and the basolateral side. But it allows water, H2O, to move into the cell and then out into interstitial space following osmotic gradients. And the idea is, if water's starting to move out, if water's moving out, you have to think, what is that going to do to the osmolarity of the filtrate? So if water's leaving, the osmolarity is beginning to increase. I'm losing the solvent, therefore I have a higher concentration of solutes to solvent. So osmolarity is solutes divided by solvent. So the amount of dissolved things per unit of volume or unit of fluid. So if I'm losing fluid, if the fluid is going down, overall the osmolarity is increasing. Osmolarity will be increasing. So the osmolarity is increasing in this region. This descending limb will flow into what's called the ascending limb. And the ascending limb has two distinct regions. One of them is the thin region and the thick region. I'm going to kind of ignore that for simplicity. But this ascending limb is also made up of epithelial cells. So imagine there's like a bunch of cells making up this border right here. And what they do is they're pretty impermeable to water. So water has a really hard time moving across the epithelial barrier that makes up the ascending limb. So no water moving across here. But they have a bunch of channels on both the apical surface, so the surface that's facing the lumen or facing the tube, and then also on the basolateral surface, which is the surface of these epithelial cells that's facing the interstitial space of the medullary region of our kidney. And what this allows for is these cells actively pump ions from the space within the tube into interstitial space. So if I'm pumping sodium, potassium, and chloride, two chloride usually are moving through these channels. If I'm pumping these ions actively into this space, you have to think, what am I doing to the osmolarity in this space, but also within this space? So what am I doing here and what am I doing here? If I'm pumping ions into the interstitial space, I will be increasing the osmolarity here, because I'm increasing the number of solutes. If I'm removing solutes from this region, I will be decreasing the osmolarity. And so this ascending limb is also known as the diluting region, because what I'm doing is I'm removing all the solutes. Therefore, all I'm left with is largely solvent. So I've diluted the fluid within these tubes. Therefore, it's called the diluting region. But within this region, I'm increasing the osmolarity because I'm pumping out a bunch of ions. So imagine all these little red guys are ions that I pumped out. And I'm starting to increase the osmolarity of this space that's located in the medullary region or the middle of my kidney. So this is all like interstitial space right here. It's located on both sides, all in here. All of that is interstitial space of the medullary or the middle region of my kidney. And I'm starting to increase the osmolarity because these pumps are always pumping ions from the space within the tube into interstitial space. But if I'm increasing the osmolarity here, and this region, this descending limb right here, is permeable to water, water will tend to move following an osmotic gradient towards a region that has a higher osmolarity. But if more water leaves here, more water's leaving, now I'm increasing the osmolarity of the descending limb because I'm losing the solvent. But that increase in osmolarity will go right over here, and I'll pump all those ions back out. So overall, this is called a countercurrent multiplication, because what I do over here on this side will affect what happens over here, and what happens over here will affect what happens over here. So it's a loop. And over time, it'll increase the osmolarity within this medullary region, depending on how far down these tubes go and the amount of channels that are present. So in some organisms, these are really long. They go really deep into the medulla of your kidney, and those organisms have a high concentration of what are called juxtamedullary nephrons, which are the amount of nephrons that actually go deep into the kidney, whereas we tend to have a lot less of those. I think upwards of 15% are juxtamedullary. So we only really have some capacity for making concentrated urine, but there's some species, especially desert-dwelling mammals, that have really long loops of Henle, and they have a high density of their nephrons are ones that have those long loops, whereas we have less. So what we've done is we've increased the osmolarity of this region. We've decreased the osmolarity of the filtrate that's leaving the descending limb. It'll then move over through this region right here, which is called the distal convoluted tubule. And from the distal convoluted tubule, that fluid that was dilute will continue out unless it is reabsorbed. And reabsorption is a bit of a long story, but the overarching theme is if a specific hormone is present, I can put aquaporin channels in this membrane, and it will allow all the water that's present in this convoluted tubule that will be urinated out eventually to move through these aquaporins. So I'm reabsorbing water when I need it. And when I need it is during a time when arterial volume is low, when arterial pressure is low. And that's initiated by these cells right over here. So there's these cells called juxtaglomerular cells. So they're cells that are located near the glomerulus. And what they are they're modified smooth muscle cells that detect changes in blood pressure on the arterial side. So remember, this is an afferent arteriole. It's coming from the renal artery, which is bifurcating off of the aorta. So it has a direct representation of the arterial pressure. And you have a lot of these nephrons in your kidney. So all of them are kind of calculating whether or not there's a lot of pressure or there's a little pressure. And if there's a lot of pressure, what it does is it stretches this little arteriole and it prevents these juxtaglomerular cells from releasing a signal molecule. But let's say that there's not very much pressure and there's not very much stretch. So this is indicating not very much stretch. This would be indicating more stretch. If there's not very much stretch, that indicates there's low arterial blood volume. And these cells release a signal molecule called renin. And renin isn't an ordinary signal molecule. It's actually an enzyme that is released directly into circulation. And what it does is it functionally cleaves an enzyme that's released by the liver. So the liver is releasing an enzyme or a protein, a plasma protein, into circulation all the time called angiotensinogen. Angiotensinogen. And renin will cleave off a few amino acids from this big protein to form angiotensin 1. And angiotensin 1 is in circulation, and all the blood in the body will eventually move by the lungs. So the lungs are a good vantage point for having an enzyme that converts this angiotensin 1, which is relatively inert physiologically, into a more potent signal molecule called angiotensin 2. So there's an enzyme within the capillaries of the lungs called ACE, or angiotensin converting enzyme, that converts angiotensin 1 into angiotensin 2. And angiotensin 2 has broad systemic effects. One of those is it goes to the brain. So it's in circulation, it's having an effect on the brain, and it's initiating magnocellular neuroendocrine motor neurons. So these neurons are originating in the brain, and they're going and they're releasing a hormone into circulation, but it's causing those cells to release vasopressin. And vasopressin is a hormone released into circulation that will go and it will come over here to this distal, or this collecting duct over here, and it'll cause the cells, the epithelial cells, so imagine these are the epithelial cells right here, making up the epithelium of the collecting duct. And it causes them to start expressing or moving aquaporin channels into the membrane. So this allows the collecting duct to be permeable. If vasopressin is absent, you won't have any aquaporins present along this membrane. Therefore, you won't have any reabsorption of water from the filtrate into interstitial space. So vasopressin initiates the reabsorption of water from the collecting duct by initiating the presence of aquaporins present on the apical membrane of those epithelial cells, which allows for reabsorption of water. And why would reabsorption of water be beneficial in a situation where we have low arterial blood pressure? Because that increase in water can be reabsorbed into the peritubular capillaries and eventually increase arterial volume, so we can counteract the initial issue we ran into over here where we had low blood volume, low blood pressure. We're reabsorbing water in order to increase blood volume and increase blood pressure. And so vasopressin also has effects on smooth muscles throughout the body that express vasopressin receptors. And there's different subtypes of vasopressin receptors. The ones that are expressed on this epithelium are V2 vasopressin receptors. They're G protein link receptors. It's a cyclic AMP pathway. But these vasopressin receptors are also present on smooth muscles, and it initiates the contraction of smooth muscles that are wrapped around arterioles that supply capillary beds. And if I squeeze on those smooth muscles, I decrease the flow of fluid moving from the arterial system into capillary beds and eventually into the venous system. And if I decrease flow from arterioles or the arterial system to the venous system, I'm overall increasing the amount of volume in my arterial system, which is counteracting this initial issue where I had decreased blood volume. And so angiotensin II will also go and bind directly with smooth muscles. So smooth muscles will express angiotensin II receptors, and if angiotensin II binds to those receptors on smooth muscles, it'll cause them to constrict, the same logic that we just talked about. Angiotensin I will also promote clotting, so it has prothrombic effects, so increase in clotting, which will decrease the amount of fluid that's being lost from the arterial system. Therefore, it's helping this situation that we dealt with before. And so overall, these are a lot of different pathways by which angiotensin II aids in both the reabsorption of water and facilitating an increase in arterial blood volume. But it also it acts as a tropic hormone, which is a hormone that releases another hormone. So angiotensin II in circulation will cause the adrenal cortex to release a hormone called aldosterone, which is a steroid hormone. And steroid hormones primarily act in terms of regulating the transcription and of genes. And this aldosterone in circulation will bind to aldosterone receptors that are present in the cells on the distal convoluted tubule, and it'll initiate these cells to start putting in more sodium-potassium pumps in the membranes of those epithelial cells. This is a little inaccurate because I'm not drawing the epithelial cells. But what happens is I pump 3 sodium from the space within the filtrate into interstitial space, and then I pump 2 potassium from the interstitial space into the filtrate. So overall, I'm pumping more ions out here. And this region, this distal convoluted tubule, is slightly permeable to water, which allows water to move with an osmotic gradient following the osmolarity. And so I'm reabsorbing water there, I'm reabsorbing sodium, but I'm also getting rid of potassium. So aldosterone is involved in the secretion or excretion of potassium, and it's also involved in the reabsorption of water, which is helping the initial problem we ran into over here, where there was not enough blood volume. If I reabsorb water here and here, I'm increasing blood volume in my arterial system. So this is just some general physiology on the kidney stuff for mammals.